Thank you very much, everyone, for joining us. Today, we're going to be hearing from Dr. Jodi Carter, Dr. David Ting, and Dr. Jake Estes. And they're going to be sharing their perspective on the spatial omics space, starting with individual presentation. And then we will regroup and have a panel discussion.
Thanks very much for the invitation to share some of our work. We've been focused primarily on these novel and very exciting high-plex spatial platforms, specifically in the context of a direct translational application of clinically actionable biomarkers in triple-negative breast cancer. So as a breast pathologist on my clinical service, I spend an awful lot of time signing out breast biomarkers. And really, until recently, they focus primarily on estrogen receptor, progesterone receptor, and HER2. While the majority of cases have one or more of these positive, there is a significant subset of breast cancers that are so-called triple negative, meaning they lack any of those clinically actionable targets. They tend to be more aggressive, the patients have a poor prognosis, and until quite recently, there really have not been targeted therapeutics available for these patients. What's changed in the past few years was a few critical clinical trials that came out starting in 2018, demonstrating that targeted therapies against the PD-L1 immune axis did show significant, albeit modest, clinical benefit in these patients. This really represented a paradigm shift in how we can approach triple-negative breast cancer and really started aggressive studies toward better understanding the immune microenvironments. What that meant from my perspective, as someone who works in the lab and signs out biomarker assays all the time, was really a massive upswing in demand for these companion diagnostics, which were determined by the FDA as being required for therapeutic approval. The issue, of course, is that these were determined by the partnering pharmaceutical industry and really hadn't been carefully vetted on the clinical side. So what you can see here, which are two consecutive sections from a paraffin block of breast cancer metastatic to liver, is that depending on the companion assay, which is just conventional immunohistochemistry, single plex, the difference in the stain intensity, because they use different antibodies with different epitope targeting, and also the heterogeneity of staining across the tumors. So unlike in other solid tumor systems where PD-L1 is expressed primarily in the tumors themselves, in triple-negative breast cancer, they're expressed in immune cells and varying subsets of immune cells. And so it became very clear very quickly the assays were not interchangeable. They do not have a great inter-observer reproducibility, and they really lack nuanced biologic insight into what it means to be a PD-L1 positive tumor, so to speak, and thus better inform patient selection. So what we did was leverage our very large, very well characterized triple negative cohort here at Mayo Clinic, which has over 600 patients in it. But for this study in particular, we focused on just under 300. Ran the clinical PD-L1 assays, scored them, used that information to target with the genomics platform using the protein modules. These data are from a couple of years ago before we had transcriptome available. So what you can see in the lower right panel is our segmentation strategy with genomics. Very straightforward. We were targeting the intraepithelial tumor segments, the breast cancers, with their immediately adjacent stromal microenvironments. When we did this, as you would expect, and shown in the heat map in the upper right corner, there is a specific set or module of proteins that are associated with PD-L1 status in the tumor. That's not unexpected. As I've mentioned, PD-L1 is expressed by immune cells in these tumors. But of course, what we wanted was to get a better appreciation of what those proteins were, how can we functionally compartmentalize them into different immune cell types, And from a quantitation perspective, which is an advantage, of course, with the genomics platform, is to get a sense of what other proteins or immune targets, ideally actionable ones, show the highest level of abundance in a relationship with PD-L1 protein. And what we can see on the right, these are data published earlier this year, is that IDO1, another immune checkpoint protein, shows the most robust association with PD-L1 protein abundance. And then we see a descending set of immune targets enriched both in antigen presentation as well as some lymphocytic subsets. So this is intriguing information, of course, and informs that clinically actionable decision-making. But of course, it doesn't tell us which cell sets are actually, rather which proteins are localizing to which cell subsets. We did harness the plex, so to speak, which is to build these composite scores that reflect immune function. So those are available, of course, from Nanostring, but you can build your own as well. And so we did that with antigen presentation, with some T-cell activation, and we built in some of our targets to then go back and look at survival data and had some very interesting observations around the spatial context of these proteins, which is, of course, what the platform provides. And it was only in the intraepithelial, or that tumor-rich segment, decided by its cytokeratin status and the intercalating antigen presentation, it was only in that segment where we were seeing significant associations with recurrence-free survival in the cohort. Now, these pairing of the modules with individual proteins were true for some targets like PD-L1, but that was not true when we looked at others, which seemed to be not, well, both spatially agnostic, but also agnostic with relationship to these functional modules. So very intriguing data, but of course, always we're trying to push the envelope and go after higher resolution single cell. So we were thrilled to have the opportunity to run this same cohort, this triple negative cohort at TMA, using the CosMx technology. So again, spatial is a big word. It means a lot of things to a lot of people. For our perspective, with the RNA reagent panels in CosMx, we were after single cell resolution, not so much subcellular. It's a different story, of course, for protein. And when we get our hands on that, we really want to see how it performs in the subcellular space. So what we did, and these are select cores from the TMA of that large triple negative cohort, is we applied CosMx, and here you're seeing a very compelling animation of how you can map the UMAP back into the spatial context. We'll see it again here from the very beginning. So you get the UMAP with the big yellow nests of tumor, and then the other colors representing specific immune cells, and then you see them map specifically back to those areas in the cores. We superimpose keratin to keep the pathologist, myself, happy that things are typing as we would expect them to do. And then we get the confidence that not only is the single cell RNA-based typing working, but that the mapping is working. So once again, as a reminder, what the actual immunofluorescence cores look like. So that's the first level of demonstrating fidelity in terms of whether those cell typing approaches actually work. And then to be thoroughly satisfied, not only typing, but with the segmentation approach, what we elected to do was superimpose the transcripts from a variety of cytokeratins, which are variably expressed in triple negative breast cancer, and illustrate this sharp delineation even further. And then you can see here on the right at a higher magnification, really nice high fidelity segmentation. So it looks really good. We're really getting the targeted population that we want, which was an important quality check for the instrument. So our next goal was to not leave it there, but to recognize that PD-L1 conventional immunohistochemistry, as shown on the right, is entirely spatially context dependent. It's not enough for the slide to be brown. You need to know what cells are expressing it in order to get a positive result. And so we designed spatial niches that replicate that clinical assay: tumor, interface, and then almost in a targetoid or radial array outward, stromal immune populations, where really it's the tumor and the interface that count, so to speak. So the application of CosMx is of particular interest to address this question, because not only can you do cell typing, of course, but there's an imposed cell state and functionality, but also cell-to-cell interaction, which includes 100 unique pairs, but also immune checkpoints. And that's really what we're after from that cell-to-cell interactome approach. And then, of course, another key thing here is something that we can't do at the protein level, which is go after the chemokine and cytokine profiles at the RNA level. And so just showing some preliminary data. Unfortunately, I can't share the transcripts. They're a little too exciting. But when we go after PD-L1, or sorry, just macrophages here now by niche, what we see from the rightmost column toward the left on that heat map is a very strong gradient of a specific subset of genes, what you would expect, some enriched in chemokine signaling, as we see on the right, in that tumor population of macrophages, as well as in the interface. And we're just showing one example here, chemokine 14, which of course has an established role in macrophages. Now, the next step, and what is really exciting about this technology as well, is we apply cell typing, right, with the RNA sequencing, or sorry, the RNA data, expression data. Then we apply the niches that I mentioned, which is critical for this. We need the spatial context to take this back to the clinical arena. Then we select out the macrophages that are in that particular niche, because again, those are the only cells of interest, or the first cells of interest to start with. And then we superimpose on those green macrophages where the PD-L1 positivity is. We do an internal control check, which is me as the pathologist going back and looking at the clinical reads from these cores and making sure that the RNA is picking out the appropriate cell population. We verify that, in fact, it has a very strong association with the clinical assay, so it's performing very well. And then we do a whole series of differential expressions, and we do them iteratively. This is just the first stop on the pathway. We're looking at PD-L1 expressing macrophages by spatial context or niche. We're comparing them to each other to determine whether that spatial location is actually critical clinically or not. We elevate that to the next question, which is, we know PD-L1 can also be expressed in dendritic cells. We know it's expressed in lymphocytes and in a rare subset of breast carcinomas. We explore all of those same relationships in all of those other cell populations from the same data set. And then we get after the really intriguing questions, which is, how do those PD-L1 expressing macrophages or immune cells of interest alter the tumor cells that they directly interact with? It's really an unprecedented level of nuance and high resolution about what it means to be a PD-L1 positive tumor, to really help direct future combinatorial therapies, as well as just give us some high importance biologic insight into the functionality of this signaling axis. And then what really is like a Pandora box for this, and is probably the most impressive aspect of all of it, is then you wipe the board clean and you start again. Rather than targeting PD-L1, you target whatever your biomarker of choice is, because it's the same data set. And iteratively, you go after all of these interactions. It really is quite extraordinary. I'll stop there. I'd like to acknowledge both the large team at Mayo Clinic, including my partner in crime, so to speak, Aubrey Thompson, who I work with on every one of these spatial platform experiments, as well as Nanostring, the technologies team, who has been very highly involved in this particular experiment. Thanks very much.
Well, thank you for the opportunity to present our work on SIV and by extension HIV reservoirs at this Spatial Genomics Symposium. To begin, it's important for me to provide some key background information on HIV infection. There are currently over 37 million individuals infected with HIV worldwide, and over the more than 40-year HIV pandemic, an estimated 37 million individuals have died. The advent and current continued research and development on antiretroviral medications has been a game changer for people living with HIV and has transformed HIV from a death sentence to a manageable chronic disease for most individuals. However, antiretroviral therapy requires daily use, strict adherence, and has side effects associated with stigma and comes at a high cost. Importantly, while significant strides have been made to bring antiretroviral therapy to people living with HIV, currently only approximately 75% of HIV-infected individuals worldwide have access to antiretroviral therapy. Thus, for these and other reasons, an HIV cure is highly needed. It's important to note that HIV eradication or cure cannot be achieved by art alone, as viral reservoirs persist under long-term suppressive antiretroviral therapy and remain the major impediment to eradication. HIV reservoirs consist of infected cells that can be either completely transcriptionally latent, as shown here on the upper left, transcriptionally active, shown here in the middle, or translationally active, as shown on the right. HIV reservoirs can also consist of uninfected cells like follicular dendritic cells, or FDCs, shown here on the upper right, that trap and retain infectious virus in the form of immune complexes. Follicular dendritic cells reside within B-cell follicles of secondary lymphoid tissues and are a relatively understudied uninfected HIV reservoir. Importantly, these cells harbor more than 90% of the virus throughout the body prior to antiretroviral therapy, as shown on this lower right image, with all of the red signal found within the B-cell follicles trapped on follicular dendritic cells. Early work has demonstrated that CD21, or complement receptor 2, is important for HIV retention within B-cell follicles in vivo, and that blocking this molecule with an antibody or knocking it out in mice reduces and prevents HIV virion trapping, respectively. Further work by Michael Carroll's group showed that surface-bound HIV on follicular dendritic cells is not degraded, rather are kept in a protective endosome and are cycled back to the FDC cell surface periodically. They also showed that FDCs can retain infectious viral particles during long-term suppressive ART, and that soluble CD21 fusion protein, or monoclonal antibodies against the C3d binding site of CD21, can prevent HIV deposition and displace bound virions from FDCs ex vivo. In partnership with Michael Carroll and our pharma partners at ViiV GSK, Rick Dunham and Heather Madsen, we sought to answer the question, can anti-CD21 treatment attenuate the establishment of FDC HIV reservoir within B-cell follicles? Because we cannot do these types of experiments in humans, we turn to the non-human primate model because of their highly similar physiology, immune systems to humans, and because SIV infection of rhesus macaques highly recapitulates HIV infection in humans. We performed a proof of principle study in which non-human primates were infected with SIV and treated with an anti-CD21 or control antibody at the time of infection and monitored the progress of infection over the first 8 weeks. Chi Chan was the lead scientist in my group directing this work. The take-home message for this talk is that anti-CD21 treatment profoundly attenuated the deposition of SIV on follicular dendritic cells within the B-cell follicles as early as day 14 and throughout the 8-week study that I just presented. To understand in greater depth the specific impact of this treatment on B-cell follicles and on SIV-infected reservoirs, we partnered with Nanostring to perform spatial transcriptomic analysis, first using the GeoMx digital spatial profiling platform for tissue microenvironment and cell neighborhood analysis, followed by CosMx spatial molecular imaging platform to perform a targeted analysis at the cellular and subcellular level, comparing individually infected and uninfected cells. We sought to understand the transcriptional differences within the larger B-cell follicle tissue microenvironment, shown here on the left, and within smaller cell neighborhoods surrounding infected cells, shown here on the right, and uninfected cells that were directly adjacent to these infected cells, in the paracortical T cell zone microenvironment. Using the Genomics Transcriptome Atlas, we collected release barcodes from 12 B cell follicles and 6 SIV infected and uninfected cell neighborhoods from each of 2 treated and control lymph nodes 8 weeks post SIV infection and post-treatment. These images are examples of B cell follicles that we captured sequence data from. And what we saw was dramatic differentially expressed genes within the B cell follicles from treated compared to control animals. Treatment resulted in a large array of substantially downregulated genes associated with a multiplicity of inflammatory and adaptive immune pathways, as well as a smaller array of upregulated genes associated with cell migration, proliferation, complement, and B cell signaling, shown on these volcano plots. When we looked at the SIV infected cell neighborhoods and uninfected cell neighborhoods, we noted that treatment did not impact transcriptional profiles, so we combined data from our treated and control animals in the downstream analysis. We found striking differences in the differentially expressed genes in SIV infected compared to adjacent uninfected cellular neighborhoods, shown on this heat map on the left. Consistent with some previous work that we performed on completely different animal cohorts and human patient cohorts, we found increased IL-10 RNA expression levels were elevated in SIV infected cell neighborhoods compared to SIV uninfected cell neighborhoods. Importantly, IPA analysis demonstrated that IL-10 may play a central role in differentially expressed genes within SIV infected cell neighborhoods. While the Genomics DSP provided robust data on the transcriptional landscapes of the B cell follicle tissue microenvironment and SIV infected and uninfected cell neighborhoods, we sought next to compare the targeted transcriptional profiles of SIV infected and uninfected cells at the single cell and subcellular level. So we utilized the CosMx SMI platform on subjacent sections from the same and additional animals used in our genomic study. Using the 1000-plex Nanostring SMI panel with 9 custom probes targeting SIV genes, we collected data from overlapping fields of view collected in our genomic study. We performed cell segmentation and SMI analysis, generated UMAP projections using a negative binomial hierarchical clustering model, and from this data set defined 14 unique cell populations within these lymphoid tissues. Importantly, SIV transcripts were dominantly found within CD4 positive T cells, the principal SIV and HIV target cell, and associated with B cells and likely follicular dendritic cells found within the B cell follicles. We can utilize spatial genomics visualization tools to see where cell populations found in our UMAPs reside spatially within tissues. And then from these cell populations, we can define tissue microenvironment niches and project those back on our UMAPs to look at those niche populations. And finally, we can identify which cell populations have SIV transcripts in our UMAPs and resolve where they spatially localize within these tissues. Furthermore, using 3D reconstruction of cells with SIV transcripts, we can delineate cells within the B cell follicles that have virus bound on their cell surface, both follicular dendritic cells and B cells, shown here on the left, and distinguish these from cells that are productively infected with numerous SIV transcripts throughout the volume of the cell, shown here on the right. Quantitative analysis of our CosMx SMI data revealed a dramatic reduction in the number of SIV transcripts, i.e. viral particles, trapped within B cell follicles in our treated compared to our control animals. Importantly, this was in striking agreement with our RNA scope and cytohybridization analysis, shown here on the right. Importantly, we also found in this analysis that treated animals had significant reduction in the number of productively infected cells within each of the lymph node tissue microenvironments. Finally, cosmic analysis provided for the first time transcriptional profiling of individual SIV-infected cells within the B cell follicle, shown here on the left, and the T cell zones, shown here on the right, tissue microenvironments, and is able to directly compare their expression levels and patterns to uninfected cells in proximity to these infected cells. This slide provides a visual example of several differentially expressed genes increased within SIV-infected cells, shown here in the middle, with this large array of SIV transcripts, compared to uninfected cells, and in contrast that to genes increased in uninfected cells in either direct contact or close proximity to the SIV-infected cells. What I hope I have shown you today in the setting of infectious disease research is that both the GeoMx DSP and CosMx SMI platforms provide complementary and rich spatial data sets, leading to a comprehensive understanding of the transcriptional landscapes within tissue microenvironments, cellular immune neighborhoods, and individual cells. And that these spatial platforms hold promise to identify specific therapeutic drug outcomes, unique inflammatory and immune biomarkers or signatures associated with pathogen persistence, and new pathways for targeted interventions to eliminate these cells in HIV cure strategies. I'd like to thank you for your attention and also thank all of those that participated in this study, both at OHSU, at Boston Children's Hospital, University of Washington, our pharma partners at ViiV, and at Nanostring, and also are grateful for the array of funding that we have received to do these studies. Thank you.
Well, thank you so much for having me here. My name is David Ting. I'm the Associate Clinical Director for Innovation at the MGH Cancer Center. And today I'll be telling you about the work that we'll be doing on repetitive elements, the Repeatome, and how they're shaping the microenvironment of pancreas cancer. So what is the Repeatome? Well, it's actually a global genomic program in human biology. Repeats comprise the majority of the genome. In fact, approximately 59% of the entire human genome is made up of these repetitive elements. And when you compare these to the coding genes only make approximately 1.5% of the entire genome. And so it really begs the question, what is all this junk DNA actually doing? And over the years, what we've learned is that in embryonic development, and developmental biologists have found that these repeat RNAs are essential for early development in model systems. We have found that these repetitive RNAs or these repeat RNAs are reactivated in early cancers, and this is an example of pre-neoplastic Pannin lesions in pancreas cancer. And work with Ben Greenbaum at Sloan Kettering, we've shown that these things are activated in chronic viral infections. And finally, others have shown that these things are important for neurodegenerative diseases. So the point in showing this is that this is not a field that is just in cancer, but seems to encompass a lot of different areas of biomedicine. And this is just an illustration of a number of publications over the last decade, including ours around 10 years ago, showing that these repeats in cancer are moving and there's some cancer-specific effects. But more recently, we've seen that there are these immune-related effects that I'll talk about in this presentation. And one of the first things we noted with Ben Greenbaum's group is that these repeat RNAs look like viruses. And this is what they're calling viral mimicry in cancer. And this is just some data indicating that the sequence composition of these repeats shown in red, in particular this HZ2 repeat, has a very high CpG bias compared to the rest of the transcriptome. Now, why is that important? Well, it turns out that these CpG motifs are directly stimulated by pattern recognition receptors, and this can engage an innate immune response. And that's what we see over here with the expression of IL-12 at the protein level. And these things look like viruses. Interestingly, they also replicate like viruses. They actually use reverse transcription just like HIV does, and replicate through an RNA to DNA conversion and reinsert themselves in the genome. In this case, the line 1 repeat is the major retrotransposon in the human genome. Finally, more recently, we found that these repeat RNAs travel like viruses, and so extracellular vesicles have been shown to be produced by cancers and have a wide array of effects as a way of delivering information to the surrounding cells that are responding to the tumor. And this is just RNA sequencing of pancreas ductal adenocarcinoma extracellular vesicles, identifying that there are these very high repeat RNAs shown here in red in the extracellular vesicles compared to the cells that they came from, indicating that not only are these repeats sensed like viruses, replicate like viruses, but they actually move to other cells like viruses. And so the cancer that we focus on in the lab is pancreatic ductal adenocarcinoma, or PDAC. This is the most aggressive cancer. It will be the #2 killer of all cancers in the United States in the next 5 years. And our 5-year overall survival for this cancer is still in the single digits despite advances in modern chemotherapy and surgical techniques. And a lot of this has to do with the fact that these tumors are very large at presentation and invading other tissues and organs. And when you look under the microscope, what we find is that, in fact, the number of tumor cells in these large masses are mostly composed of stromal cancer-associated fibroblasts, or CAFs. And what you can see here is these tumor cells in blue make up these little islands, but they're actually in this large ocean of stromal CAFs shown in red. And work done by David Tuveson's group showing by single-cell RNA sequencing that these cancer-associated fibroblasts in pancreas cancers are actually different subtypes. There are different types of these fibroblasts that we see with heterogeneity in these tumors. And the inflammatory CAF, or ICAF, and the MyCAF, the myofibroblast CAFs, are the main two CAF types that we find consistently across our models in pancreas cancer. And our work a few years ago that we published indicating that cancer-associated fibroblasts, when mixed with pancreas cancer cells, can induce sort of more aggressive phenotypes with EMT as well as proliferative capability. One of the things that we found in the same paper is when we looked at the fibroblast co-culture with the cancer cells is that they actually induce an interferon signature as well as proliferative signatures, indicating that the fibroblasts almost as active as if they have been infected by a virus. And so that led to this question: are cancer repeats RNAs in these extracellular vesicles a mechanism to induce this CAF heterogeneity and cause an inflammatory type CAF? And so what we did was we took pancreas cancer extracellular vesicles, or EVs, and we treated these fibroblasts in tissue culture and then purified those RNAs and looked for interferon stimulated genes, an indicator that they are responding as if they're infected by a virus. And when we did that, as you can see here, for 2 different pancreas cancer exosomes or extracellular vesicles, we could see the induction of significant levels of these interferon response genes, or ISGs. So now that we have some functional data showing the promise of this phenotype, the interesting thing we wanted to do next was, can we see this spatially in tissue? And what we're showing you here is a collaboration with Nanostring, where we performed spatial transcriptomics on a human pancreas cancer tumor. And each of these fields of view in these yellow boxes is an area that we're focused on based on where a pathologist has told us this is an interesting area to look at. And that's one of the main strengths of doing this type of spatial analysis with this type of platform. And you can do the traditional things you do with single-cell RNA sequencing. You can do the UMAP technique to sort of separate into clusters. And what you can see here are the tumor cells are nicely clustering over here in this green area, and the immune cells and other sort of cells are over here. And when you start to subcluster, you can start to see that there are different tumor subclusters, as well as in this immune compartment, there are some fibroblasts that are there, as well as different immune populations. Now, the beauty of this spatial technology is that now you can take those UMAP cell type clustering phenotypes identified by RNA-seq type programs, and then map them back onto the tissue. So you can actually see histologically where are these cells living. And you can see all the different neighborhoods that you can see across these 4 different tumors that we analyzed with Nanostring. And if you zoom in, we can now start to ask the question, are these repeat RNAs in different locations in the tumor and spreading away from the tumor? And in this case, we've made custom probes with Nanostring, including these 5 different repeats. The line 1 ORF1 is the main one that is expressed at highest levels across cancers. And what you can see with these yellow dots is we can see these tumor glands that are positive, but also some positive cells that are moving out from those glands. And when we focus on individual cells and phenotype and quantify, what you can see here in purple are the line 1 repeat RNAs elevated in the tumor glands. And as they're going away from the tumor glands, there's like a gradient that you can see going into the stromal microenvironment. And now we can CAF subtype all of these things, where you can see the ICAFs and the MICAFs with this type of information to sort of see how do the MICAFs and the ICAFs change in the setting of these repeats, seeing them. And the important thing is it's not just an N of 1. You're not just looking at one part of the tumor, and you're not just doing some deep analysis there. This type of technology, you can look at multiple fields of view in the same patient, so looking at intratumoral heterogeneity, and you can look across patients. And that's a powerful thing for us on the clinical translational side to really see how do these repeat RNAs relate to the CAF relationships that we're seeing within a tumor, but also across different tumors. And so in summary, these repeat RNAs are expressed in their aberrant in many cancers as multiple levels of this viral mimicry. These repeats can be transmitted to surrounding cells through extracellular vesicles and stimulate an interferon response as if they're infected. And spatial transcriptomics provides an integrative way of seeing these cell phenotypes mapping with the repeat RNA localization to provide really novel insight into the biological phenomenon of this in actual human patient tumors. There's so many people to thank. This is really led by Yune Yu and Rebecca Porter from my lab, as well as my entire team. Of course, our collaborators at Nanostring, and the many others who are involved with this project. So thank you so much for your time, and look forward to the panel.
Thank you very much for this insightful and exciting presentations. What we'll do next is have a quick Q&A sessions to discuss in more depth some of the insights you get from these platforms. So thank you very much, everyone, for being here. I'll start by asking the first question to David. And David, I'd love to hear about how you keep track of all of the developments in the space. Obviously, the space is moving really rapidly. And I'd like to know, how do you track these developments and which one are you most excited about?
Yeah, that's a great question. Well, fortunately, being in Boston, we are in this nexus of Harvard, MIT, and Broad. So all the latest technologies of different types sort of enter into our ecosystem in one form or another. And I've been fortunate that I've done all my training here since I was a little undergrad. And so I get most of my information from my friends. And I think the technologies out there, there's so many in the spatial transcriptomic area. And I think the promise is really, the technologies that can take human archived tissue material and really do this type of analysis, because that takes it from just something that you can do with experiments with mice or occasional patient samples to deploying it across all biospecimens in our major hospitals.
So Jodi, next question to you. How do this innovation enable you to think differently about your research?
Yeah, really great question. And a good follow-up actually to what my colleague just mentioned. As a pathologist who specializes in biomarkers and reads them out clinically all the time, we really use antiquated spatial technology, right? We use conventional immunohistochemistry that's been around decades, if not a century at this point. So it's very nice to see these novel technologies being applied to conventional clinical samples. And what really excites me about that as someone who does biomarker work is being able to, harness the plex, so to speak. These technologies are really ultra high plex, not just in the transcriptomic space, but for protein. And really that's where the action is, so to speak, in my area, which is clinically targetable biomarkers, which by and large are protein. So it's really an exciting time to be a biomarker clinician.
Nice. And we'll touch on that a little more during our discussion today. Certainly about talking about clinical samples. It's going to be interesting to unpack that a bit. Similar question to you, Jake, and thanks, Jodi, for teeing this up, is what type of questions can you answer now that you have access to this platform that have such high plex and high resolution, coming at it from a slightly different angle from David and Jodi on the oncology side, obviously you being more on the infectious disease side. Can you comment on what you can get with this cellular or subcellular level of resolution that could not really be addressed before?
Yeah, thanks for the question. I think that from my perspective, there are really a wide range of questions that you can now answer using these spatial transcriptomic approaches and spatial profiling approaches that were simply not possible before. One in particular in the field that I study, HIV infection, is really to fundamentally understand the transcriptional profile of infected cells, which we've never been able to do either outside in a test tube or within, certainly within this spatial context in which these viral reservoirs reside. In addition to that, because these cells don't function and interact in isolation, we fundamentally want to understand how these infected cells are interacting with their cellular immune neighbors to identify potential therapeutic interventions that would help us to clear the body from these pathogens. So it really, I think, opens a door for both a fundamental understanding of the biology, the immunology of these infectious disease, but also as a discovery tool.
Excellent. All right, so what I'd like to do next is actually take it like one level deeper and better understand how these technologies have enabled you to, has affected your research essentially personally. And maybe starting with you, David, can you comment on how you've been using these tools from Nanostring to better understand gastrointestinal cancers, or other type of cancer and how it has impacted your research?
Yeah, absolutely. I mean, I think one of the things that plagues us in cancer is that it's a multicellular mess. And one of the things that I've focused on as an oncologist is how do we find new drugs and biomarkers that can help us deliver the right types of therapies to our patients. The things of the past are single cell RNA sequencing. We were able to refine our single cell profiles and sort of identify different subtypes. But at the end of the day, I was once told that an oncologist is only as good as your pathologist. And I think it's so true, because when you see cells that are interacting in a certain way on tissue, it tells you something that none of these single cell RNA-seq or other types of technologies can tell you. And those interactions are the ones that my lab works on in the lab to understand the functional relationships, in my case, pancreas cancer, of tumor cells with fibroblasts. And if we can understand that in the cell culture system and animal models, but ultimately find it in the actual patient specimens, those are the interactions that I would want my friends to drug. And at the end of the day, I'm here to go kill some cancer cells to help my patients and to ultimately really try to cure some of these patients with pancreas cancer.
Nice. And I agree. Seeing is believing, really much so. And I think that in this case, you can make really pretty pictures and also really delve into some very complex biology. So thanks again for also teeing up the question to Jodi next, actually very nicely talking about clinical samples. And a question that I have for you, Jodi, is can you tell us about some of the insights on clinically actionable targets that you've had using these technologies?
Yeah, absolutely. And quick shout out to David for the pathology call out. Much appreciated. As he was saying, I mean, it's a partnership, right? What I use as a pathologist is the insight from my oncology colleagues about what the emerging biomarkers, companion assays will be, so that we know what best to target and how to gain insight. So the most relevant example for my work in triple negative breast cancer is the PD-L1 axis. So targeted immunotherapy, which was a big deal, a game changer for this subset of patients. But really, as a monotherapy or with conventional chemotherapy, has had somewhat modest benefit. So what we've done is leverage using geomics and now into the cosmic space with single cell resolution, really interrogating what it means to be not just a PD-L1 positive cell in the context of these cancers, but also which are bystander cells and which are actually clinically meaningful and responsive, or else driving response to immunotherapy, to targeted immunotherapy. So that's one critical area that it's very gratifying to see some very key results, first with geomics, and then to take that a big, deep dive lower, so to speak, with cosmics. Very exciting. Similarly, in the HER2 space, so targeted therapies with HER2, and now moving into antibody drug conjugates, where it's not so much about just really overwhelming amounts of a biomarker, but rather leveraging a target to deliver a toxic payload, and how can we inform that toxic payload? But of course, my colleague David would know a lot more about that than I do.
It's actually a comment on this. It's interesting to see the interaction here. Do you believe that the increased complexity of these platforms and technology can actually enable or necessitate additional interaction between pathologists and oncologists? Or do you think that it's still going to be more limited interaction, or at least in-person interactions?
Well, I'm not sure who the question is targeted to, but from my perspective, it really is essential to have a multidisciplinary approach, right? I mean, what tends to happen, particularly in the last few years, is that, as the biomarker specialists clinically, we're almost caught unawares when the companion diagnostics emerge on the scene if we have not partnered with our colleagues who are aware of what clinical trials are, which are the most promising, et cetera, which then gives us a chance to really get on top of things, right? So it really is a functional partnership, and I hope that moving into this spatial space, which is really visual, that's what it's all about, can help facilitate those interactions and ultimately provide the best patient care.
Mm-hmm. Yeah, and just to follow up on that, I think pathologists and oncologists have been working together for decades, and that's the success of the ALK translocation story, the EGFR mutant story. I think that this technology enables a whole nother type of multidisciplinary team. And, in oncology, we always have many pieces to the puzzle to treat the patient. We have the surgeons, we have the radiation oncologists, we have the medical oncologists, we have the pathologists, we have the radiologists, we have the gastroenterologist. But the piece that has always been missing is actually, how do we integrate the biologists and the engineers? And I trained at MIT as a chemical engineer and in biology. And, in many ways, I'm not the best engineer. I'm not the best biologist, but I know how to translate between the disciplines. And I think that that's the opportunity of this instrument, these types of instruments. You can start talking with biologists about, what does this mean when this cell has all these programs and it's next to this cell? And the biologist is going to, and the clinician is going to tell them, well, in this patient population, these are the important things that we need to figure out. We need to figure out why these cells are working together to ultimately spread or to become resistant to chemotherapy. And so I think that that's the promise. You can see the future of medicine or biomedicine changing, where you have people who are in the lab even more integrated with what we're doing on the clinical side. And I think that's the exciting future for all of us.
Nice. And we'll get back to this concept of meaning and data actionability a little further in this discussion. Turning to you now, Jake, I wonder if you could comment on and share your perspective on how this technology can be used in order to really better understand cell phenotypes and the underlying biology, right? All of the molecular markers that come along with it. And as well as commenting on how it enables you to better understand the inflammatory landscape. Turn it to you.
Yeah. As a classically trained immunologist that works on, host pathogen interactions, we have had a suite of highly sensitive and quite elegant immunologic tools that have allowed us to understand the complexity of the immune system for quite some time. Multi-parameter flow cytometers, cell sorters, CyTOF, and the one really thing that's lagged is our ability to image these cells in situ in the same level of complex detail that they exist. And so for me, what's truly exciting is the ability to be able to understand at single cell and subcellular resolution the transcriptomic profile of these immunologic system interacting with each other in the context of an infectious disease or in the context of a cancer or other chronic disease in unprecedented scope and level. And the merger of the RNA's transcriptional profiling with high dimensional protein profiling, to me, is just such an exciting aspect, because you're now going from the RNA to the protein function to the immunologic outcome.
Interesting. I remember from back in the days the dogma like DNA, RNA, protein. Do you think that these platforms are gonna try to challenge that dogma a bit?
Challenge it, probably not, but access it, certainly. I mean, I work on HIV disease, and a large problem are virally infected cells that are latent. They're not transcribing viral RNA. And we have in situ approaches to be able to identify them. We help develop those approaches. If we can now incorporate those into these types of platforms that then merge with these high dimensional transcriptomic and proteomic approaches, we will have a level of understanding of these pathogens that persist in an unprecedented way.
Great, thank you. So the other thing that the three of you have in common, you may or may not know, is that all of you actually have access to GeoMx and CosMx data. It's pretty unique. And it'd be great for each of you to elaborate on what you've learned from this different data set and what's the draw to go and use a CosMx platform as opposed to GeoMx. What is it that you get out of higher resolution? And what is it that you get out of like higher plex? Like obviously the GeoMx can do whole transcriptome and the CosMx can go subcellular. Maybe starting with you, David, can you maybe comment on this?
Yeah, for me, it's really, when you're doing whole transcriptome and the GeoMx platform, you can really see depth of molecular profiles. And I think that allows you to really refine your hypotheses or generate those hypotheses that are going to be high yield when you start looking at spatial data with the CosMx. Because if you were just to look at the CosMx in isolation, you could just get lost in all the data and all the XY coordinates. But I think when you know that there's certain genes that are maybe upregulated when a fibroblast is close to a tumor cell or far away from a tumor cell, then you would have a hypothesis that you can drive down on the spatial data. And I think that's the main thing that that I've been able to sort of think about. I think the other thing is that you start to think about the other element we never think about in clinical medicine, which is time. You start to think about what do transcripts potentially mean for cells that may infer time. How long did it take this cell to turn into this cell? And that concept of plasticity is probably the root of all evil for cancer.
Interesting. I actually discussed that with someone at a sequencing company about whether it would be possible to add some kind of marker that enables you to know when the transcript has been transcribed in the cell. That'd be actually pretty cool if you could do this. Great. Jodi, same question to you. Can you tell us a bit about the tension between plex and resolution, how you think about it, and in which case you think one might be more useful or important than the other?
Yeah, so I think that, having worked with the genomics platform, primarily in protein, but also now in RNA for the last several years, uh, it's incredible what we can do, right? And we can get down to several dozen micron scale, so to speak, but you're really not getting that single cell resolution. And of course, it's always, this is wonderful, but what next? And I think that some of the intrinsic assumptions that have to be made with a segmentation approach in genomics can get answered with cosmics. But of course, the issue always is that now you've opened a Pandora's box of what do you do with that information that you're getting, and really leads to another issue that we may or may not come up. But the bioinformatic demand around this data is quite large, right? So I think that, in terms of which is the better platform, I think they're, as been pointed out, they're offering different data and they're offering different potential applications. And I think that harnessing data from both is probably the ideal way to go if that's possible. But I think because cosmics is the newest, shiniest, I'm pretty impressed with what we're getting out of that right now.
Nice, good to hear. Jake, same question to you. If you could tell us about this tension between plex and resolution, and comment on how it plays a role in infectious disease and inflammation.
I'm a huge advocate for utilizing technological approaches to answer very specific biologic questions. And so for me, I look at these different platforms as synergistic tools in order to answer very specific and unique biologic questions. For example, with the genomics, we're able to look at tissue microenvironments and cellular immune neighborhoods at really unprecedented level, looking at whole transcriptomic profiles of cell populations within a particular region, both before and after a therapeutic intervention or after an infection in a model system. But one of the really large advantages of the cosmics spatial molecular imaging approach for me is being able to actually dissect down at a single cell and subcellular resolution of the transcriptomic changes and proteomic changes within the infected cell itself, within the context of the cells that it's interacting with. And so for me, that is an opportunity to really understand how this particular viral pathogen, and that you can apply this across any other pathogen of interest, of how those particular pathogens are actually altering those transcriptional landscapes, and then how those cells are interacting and influencing those cells around them, and likewise how those cells around them are actually influencing that infected cell.
I'd love for you to elaborate a bit more on this, actually, as there is this little bug going around here that's creating havoc. And is there anything you could share in terms of how these platforms can be used in order to better understand, the essentially COVID-19 and better understand like SARS-2 infection?
The ability to actually understand key aspects of these viral pathogens or any pathogenic disease is actually having the right sample to actually analyze, right? And in many cases, those right samples come from animal models, because you're able to do longitudinal sampling or sampling in unprecedented depth that you're not capable of doing at the clinical system. And so utilizing those type of approaches, you can really understand at a level that is just simply not currently possible with other platforms, the very specific immunologic process that is transpiring early after a viral infection event, and then how that either transitions into viral resolution or viral clearance, or how it diverges into chronic inflammatory processes that lead to more severe disease. And so these types of platforms, I think, have the potential or have the promise of being able to help us understand where the helpful immunologic responses diverged from the more catastrophic ones that lead to chronic and severe disease.
Thanks for the answer, Jake. And David, I believe that you also have some experience with COVID-19 and the use of this platform. Is there anything that you can share? I know that you've played around a little bit.
Yeah, I mean, I think we've learned a lot about, in many ways, the similarities between COVID-19 and its effects on cells and how that's similar to some of the things we see in cancer. And I think that's one of the benefits that we've had about just trying to help in the situation. We were working with a variety of different partners just to, do what we could on the research side. And I think, from my standpoint, it was a trying time. Boston was hit, second after New York. I'm a medical oncologist, and, we were asked to volunteer. So I went in and took care of those COVID patients, and my lab rallied around that. And they went into the lab and said, we're going to go study COVID. And I think that that's the thing about our great institutions is you find people stepping up to meet the task of something as disastrous as a pandemic. And you realize like how great people are.
Nice. That's inspiring. So shifting gear, Jodi, I was wondering if you could comment on how should people think about incorporating spatial genomics or spatial omics into their experiments and what should be the experimental design for these experiments?
Sure. So we get this question a lot, of course, because there's a lot of interest in these platforms. And I think that, like any technology or any novel application, there's a lot of not misunderstanding, but potential for misunderstanding about the advantages and the limitations of any platform. So to just step back, the word spatial means different things to different people, right? And it's really about what is the best fit for the experimental design. And the primary question really is spatial actually required? Number one, what does spatial mean to that person as well? And then also to understand that with geomics, right, you're not getting that single cell resolution, and to be completely clear about what the output is. So if it's really a discovery-based transcriptomic approach, then of course it can be very powerful. You can have a targeted application to your, resolve or address your question of interest. But there's always that, for me as a pathologist, very visually oriented, that lack of insight into, can we assign whatever these findings are to a particular cell subset, et cetera. Mm-hmm. So I think that, information or insight into the platforms, whatever they be, is obviously essential. I think the other component here that really has to be understood before embarking on these things is the bioinformatic need is not trivial, particularly with cosmics, right? When we're getting into now single cell resolution, where we're harnessing not just this quantitative output, but also really high data imaging, right? That's really what's happening here. And in order to get the most out of that, you really need a specialized team who will know how to handle those kinds of data and extract what you can get from it, which is really quite impressive. So I think that would be just in general some considerations if you're thinking about these particular platforms. I would always say it's definitely worth it if you can do it and if it addresses your question.
Great. And a very similar question to you, David, actually. Could you comment on some of the best practices and what you've done in order to get the most out of the data that comes out of this platform? Because as Jodi just mentioned, it's clearly one of the key challenges today.
Yeah, no, I think they're still needing to be built. And I think, we've been looking at the data on the cosmics, and I think, it reminds me, and it's named, I think, probably in the same way. It reminds me of all the stars of the galaxy and looking at it. And, before you had like people that just looked up at the stars, then we built telescopes, then we built, a telescope that we sent into space And you could see things that you could just never see before. And I think, and so, I have a good friend who's a physicist who turned into a computational oncologist, Ben Greenbaum, who's over at Sloan Kettering. And, the physicists have been thinking about these problems for a long time. And I think the solutions for these types of questions are not going to be conventional things that you can do from, simple statistics, but it's going to require someone in astrophysics who's going to help us deal with this type of data. The same people that can understand why stars are in certain positions and why planets revolve around them are similar to what our questions are going to be for, cells in space and transcripts. The important thing here is also, I come back to the pathologists. They have been looking at human tissue histology for, hundreds of years, I think, it seems. And their ability to see patterns is beyond comprehension for any computer or any modern day artificial intelligence. And we still need to figure out how to see what they're seeing and then apply statistical models around that. And I think that that's the power that this can bring, again, is just bringing people with very diverse expertise together to solve very difficult problems. And from my standpoint, again, this time element, I come from engineering, so I think about flux. And one of the questions is, are cells in flux? And can you see that? And I think you can infer that with single cell RNA sequencing, but this type of technology, you can see it.
So that's fascinating. One challenge I remember talking to people in a bioinformatics space on a commercial side is that historically it's been actually very hard to get people to pay for software. And in some sense, you're competing with NIH and open source, and you're competing with free. Do you think that the increased complexity of the data that we get, it might actually start to get people to realize the importance of the bioinformatics and an increased willingness to actually pay for it, given there is so much of the value locked into this data? And that's a question for all three of you.
I could take a crack at that. As a basic scientist in an academic institution, as we all are, we have a lot of financial constraints of what we're able to do. And one way around that is really forming, really fruitful collaborations and teams that have the level of expertise that can help us address some of the issues with computational bioinformatics. I think that's, from my perspective, one of the largest challenges with wide scale adoption of these particular platforms is the access and the ability to learn quantitative analytical and bioinformatic tools that you have to be applied to your data, these rich data sets. And so I don't think it's necessarily an unwillingness to pay for those analytic tools, but I think it's some both constraints on small labs and their finances, as well as actually learning those tools themselves.
Anything to add, Jodi or David? If not, like Jake, I actually have a follow-up question for you, because we had discussed some of these challenges of adopting this platform. So in addition to the one you just mentioned, are there any additional one that you'd like to share that come to mind?
Yeah, I really think that the data analysis aspect is probably the leading challenge to wide-scale adoption of these platforms. I think in addition to that, you really do need to have at least an intermediary, if not an advanced understanding on imaging and histopathology and pathologic processes. My lab has been doing immunopathology for infectious disease for nearly 20 years, and we've trained a lot of people, and there's still not a lot of people that are doing what we do, because it's just not easy to incorporate this into your lab if you're not fully invested in it. And so those are what I see as the major challenges to wide scale adoption.
Great. And there is actually a concurrent session on the bioinformatics side that we do encourage people to go and listen to as well. And I'm sure they have a great discussion on topic as well. Moving on to the last question that we have for each of the panelists is, what do you think is going to be the next big innovation in the space? Like, what is it that you're particularly excited about? It ties back to some of the first things that we discussed, maybe starting with David.
Yeah, I think the next level of things is, how do you integrate images with image morphology features with these transcriptomic or protein patterns? And I think a lot of the artificial intelligence community is already starting to do this. In particular, I think the easiest one to think about is the nucleus. We know that open chromatin state has a certain histological feature with prominent nucleoli and other things that our pathologists have seen for years. But how do you relate that to what you see in the RNA transcripts in those same cells? We already know that they're related because chromatin and epigenetics are clearly linked with transcription. And so I think being able to connect those in a tissue-specific context, so if you're a colon cell versus a white cell, those features in your nucleus probably mean different things. And so connecting those dots, I think, is the next future. And in many ways, when you think about it, it's sort of like taking your telescope and going even further to see what's really in the nucleus.
Nice. Jodi, same question to you.
Yeah, I have a very similar answer. I think that, again, as a pathologist, and always great to hear nice things about pathology, but it's humbling to realize that, we can recognize a lot of things on tissue, but we don't see everything. And certainly with this rapid advent and even clinical implementation of AI tools, our jobs aren't in jeopardy now, but could be a different story 20 years from now. So I think that implementing these kinds of novel, but even what has now become standard AI, you can merge that with CosMx image sets, right? So if you're for RNA, sure, for subcellular localization of protein across 100-plex protein, and the number of images that you get across that 100-plex for CosMx is just staggering. You can't do that with anything else. You can't do that with conventional immunohistochemistry. You can't get that kind of leveraging that plex to get some insight about who is partnering with whom and is that actually meaningful. And I really think that, as great as we think we are in pathology seeing things, that really is going to require some AI, and that's really going to lead the way.
Agreed. That certainly makes sense. I'm also guessing it's going to be sooner than 10 years. If it grows at geometric scale, I've seen the movie. It doesn't end well.
Let me at least retire then first.
Jake, same question to you.
Yeah, I don't know that I'm great at predicting where technological advances are going. I can certainly tell you what I would be certainly delighted to see emerge. As Jodi and David mentioned, the incorporation of high-plex RNA profiling with high-dimensional protein profiling, to me, is just would open up an enormous amount of doors and answer an enormous amount of questions from an infectious disease and immunologic processes associated with those diseases. But in addition to that, how are these RNAs regulated? Seeing how we're able to see the regulation of microRNAs or even metabolomics as an incorporation to these spatial approaches, I think would really open up just rich discovery capabilities.
Mm-hmm. It's interesting. None of you have mentioned the microbiome, and I don't see a lot of people talk about the microbiome in the context of spatial biology. Any thoughts about discussion you've had or ideas that it seems like a great platform in order to understand the interaction between all these microorganisms that are living in our guts and everywhere. Anyone?
Yeah, I think you make a really compelling point. I think probably the reason we didn't talk about it because it's that's a level of complexity that is well over my head at this point. We need to focus on the easier questions like viruses, I guess. But we do study tuberculosis in non-human primate and models and in humans, and so there are multiple areas in which that host pathogen interaction or those host commensal interactions are really fundamental. And these platforms, I think, certainly would be a rich way of answering some key questions.
Well, thank you very much to each of our panelists for sharing their findings to date, as well as the Q&A that we had. I think it was very insightful. We're very much looking forward to some of the work that you'll be publishing with these platforms, as well as the way that other scientists might be using these platforms in both research and clinical settings.